We're going to be talking about translation and the genetic code. Translation is the process by which the mRNA is decoded by tRNA and translated into amino acids. In the cytoplasm, mRNA serves as a template for protein synthesis, which occurs in ribosomes. The nucleotide sequence in mRNA is read and translated into amino acids. Almost all cells use the same genetic code to convert the nucleotide sequence into amino acids. Conversion of the mRNA to amino acids requires tRNA to decode the mRNA template. Let's take a closer look at the 3 steps of translation. So the first step is initiation. In eukaryotes, the 40S, or the small ribosomal unit, binds to the 5' cap. The charged initiator tRNA binds to the AUG codon with the initial amino acid to be methionine. The large ribosomal subunit that binds to the smallest subunit creates the initiation complex. The second step is elongation. This is a 3-step cycle that is repeated for each amino acid. The ribosome moves in the 5' to 3' direction along the mRNA. The A site is this is where the next amino acid that is being added to the growing chain and is determined by the mRNA codon within the A site. The P site holds the tRNA that carries the polypeptide chain, and the peptide bond is formed as polypeptide is passed from tRNA in P to tRNA in A. And the E site is the uncharged tRNA remains until exiting the ribosome, where it becomes recharged. The final step is termination. So once a stop codon moves into the A site, termination begins. The release factor protein binds to termination codon, and the polypeptide chain is hydrolyzed from final tRNA. How do 64 different codons produce 20 different amino acids? The genetic code, which is the set of rules by which information encoded in genetic material is translated into proteins. The part of the genome that codes for a protein or an RNA is referred to as a gene. So what makes up the genetic code? A codon is made up of 3 nucleotides, which forms a unit of genetic code in a DNA or RNA molecule. The codon specifies an amino acid in a protein, and the exception is the stop codons. This code is considered to be degenerate, which means different codons specify the same amino acid. The genetic code is made up of 64 different codons, and of those 64, 61 encode for amino acids and 3 specify termination of translation. And there are also only 20 amino acids. In addition, there are start codons and stop codons, which initiate and terminate translation respectively. The wobble hypothesis was discovered by Francis Crick, who states that rules of base pairing are relaxed at the 3rd position so that a base can pair with more than one complementary base. So this is what the genetic code looks like, and as you can see, the ones that are circled, AUG is a start codon, and UAA, UAG, and UGA are the 3 stop codons. And this code is also considered almost universal. Why is all of this important? Translation allows for RNA to be processed and synthesized into proteins that are essential for our bodies to function. Without translation, our bodies will not be able to make proteins. The body makes about 3 times 10 to the 20th proteins per hour. Without the process of protein synthesis, life would be nothing like it is today.